Have you ever wondered what's in our blood? What it can tell about our well-being? What about the innovative technology used to unravel the mystery of what's in our blood? Hello, I'm Dean Whiting, and this is It's in Our Blood, the podcast series that explores the critical role technology plays in the pathology laboratory, in hospitals, and in the community setting. So thank you everybody for joining us this time around, and I'm very pleased to say that we have Dr. Gary Harton with us. He's the Director of Market Development at PerkinElmer. But more importantly, possibly, Gary's 25-year laboratory scientist with a PhD in genetics. He has some teaching positions, one at the University of Kent in the UK, one at Eastern Virginia Med School, and one starting up soon at Bryant University. So I just wanted to say a warm welcome to Gary, and maybe pass over to you. Is there anything else you'd like to add to the introduction that we should know?
No, I think you kindly introduced me quite nicely, Dean. Thanks for having me.
Perfect. All right, well, let's get on with it. So we're talking about your speciality, which is pre-implementation genetic testing, especially for aneuploidy. Now, I don't know much about this at all, so I really appreciate you explaining a little bit about pre-implementation genetic testing. What is it? And maybe you start there, and then we'll maybe ask some follow-up questions.
Yeah, sure. I think it's quite interesting, right? We've been doing pre-implantation genetic testing now for over 25 years, yet I'm convinced that the average person on the street has no idea that we own the technology to do a small biopsy on a growing human embryo and then test that embryo for a specific genetic disease, say cystic fibrosis or spinal muscular atrophy. We can test that embryo for structural chromosome rearrangements if the parent carries a rearrangement, a translocation, for instance. And then the thing that we're going to talk about today, we can also test those embryos just to make sure that they have the right number of chromosomes. I'm sure everyone's heard of Down syndrome before. You might have heard of Turner syndrome or Klinefelter syndrome. Those are all syndromes where a child is born that has too many chromosomes. So for Down syndrome, for instance, that's an extra chromosome 21. It turns out that those can be live born, and they do create problems, obviously. But what we found out over the last 20 plus years is that those aren't the only chromosomes that can be abnormal. It turns out that during development, every single chromosome you can find in an embryo either in a gain state, so having an extra chromosome, or in a loss state, having a loss of that chromosome. And what that then leads to then is an inability to get pregnant. And it's why as patients age, particularly as women age, they have less and less chance of actually successfully having a pregnancy. So during IVF, what you'll see is the average age patient hits the IVF cycle or hits the IVF center around 38 years old. Their chance of getting pregnant and staying pregnant at 38 years old without any other technology is probably in the 30 to 40%. By the time they're 4 or 5 years off from that, by the time they're 42, their chance of getting pregnant is near zero. And that's mainly, in large part, we think that's due to the fact that there's all these embryos that are chromosomally abnormal. So this testing, pre-implantation genetic testing for aneuploidy, allows us then to screen, say, 3, 4, 5 embryos that are sitting there during an IVF cycle and say, these 2 embryos are chromosomally normal. They have the chance of getting you pregnant and delivering a normal live birth. These 3 embryos that are chromosomally abnormal, we know from data, there's very clear data that says those embryos are never going to lead to a normal live birth. So let's not spend time and money transferring those embryos. Let's only transfer the euploid embryos, transfer them one at a time, and increase your chance of getting pregnant and staying pregnant, delivering a healthy child.
Wow. I mean, that's so important to people in the world who are desperate to have a family, desperate to have kids, really struggle and, go to IVF services. It's great to know that there's technology which can maximize the success of this process, of the pregnancy process. And just so great that technology can have such a big role in helping people in that way. Can you tell me, approximately how many times is this accessed? How many people are accessing this kind of pre-implementation genetic testing?
Yeah, I think, it's a great question. And I think, my answer is simply, if you think of it very simply, not enough,
Not
right?
enough, yeah.
And the reason for that, there's many reasons for that. One is cost. It's not free. IVF is expensive. Adding on testing is expensive. It's technologically advanced, obviously. We'll talk about technology in a minute, I'm sure, but you have to be able to do the genetic testing in a way that makes that's good but it can be it's next gen sequencing based, so it's not everyone has a next gen sequencer sitting there. And then lastly is just the technology. I mentioned, today to do this testing, one has to take an embryo and cut a hole in the side of the embryo and gently get some cells off that embryo while still having the embryo remain alive. And that technology, while it exists in every IVF center in Australia and it exists in every IVF center in America, it doesn't exist in every IVF center in the world.
So, since we're moving towards the technology, and the technology is really the important part here, I guess, so what is the traditional technology for studying the genetics of the embryos for aneuploidy? How do you go about that?
Yeah, so today we use next-gen sequencing. Most everybody uses next-generation sequencing. You can do that in many various different ways. There's a number of commercial kits that are available from the companies that you'd expect to make commercial kits for this kind of testing. There are some homebrew methods. There are some people that have their own methods. But almost everyone, no matter how they get to it, they create a library, they barcode the samples, and they run them on one of the big next-gen sequencing platforms. That's what we do today. Previous iterations have included array CGH, traditional CGH, and FISH before that. But today, 99.9% of the testing that goes on in the world is done through next-gen sequencing today.
Wow. And as you say, I mean, next-gen sequencing is becoming, pretty much standard technology in a lot of labs around the world. This podcast is brought to you by Pathology Technology Australia, the peak body representing the companies who invent, manufacture, supply and support the vital technology needed for testing those precious patient samples. Did you know that pathology tests provide vital information for about 70% of medical diagnosis and 100% of cancer diagnosis? Pathology Technology Australia, representing the companies who provide the innovative technology so critical to a high quality, affordable and accessible healthcare system. But we're constantly pushing back the fronts of science. So, what sort of new technology might be coming along? Say something that maybe open it up to greater access or make it easier. What is there anything new coming along in this area?
Yeah, I think there's sort of two new things. I'll go through one technologic thing and then one sort of not really technology, but just a different way of viewing this. And I think they're both quite interesting. So the technology, one of the reasons that this test doesn't happen on every cycle, as I mentioned, is cost. And so if you happen to work in a large laboratory like I've worked in, where you're testing thousands of embryos at a time, it's quite easy to envision having hundreds of samples every day, and therefore taking and harnessing all the power that a next-gen sequencing platform gives you, right? Because you can barcode, you can run 100 or 200 or 300 samples perhaps, all in the same run. Miniaturization of sequencers, miniaturization and automation in a way that makes it possible to run less samples in one run, I think long term will open up this platform and this test to more individuals, where an individual IVF center today that looks at it and says, I can't possibly have enough samples ever to buy a next-gen sequencing machine and all the stuff that goes with it. However, some of the companies have really miniaturized next-gen sequencing, where you can reliably and quite cost-effectively run 8, 12, 14 samples at a time. You can also imagine that as we continue to miniaturize the sequencing end, the library prep end, which anybody that works in a next-gen lab knows that library prep is difficult. It's time-consuming. It can be persnickety. It can be difficult. If you can miniaturize that and automate that in a way that makes sense, I think that's where the technology will go next, is into smaller sample sets that you still can harness the power of sequencing, but for a lower price point than we could, let's say today or a year ago. I think that's one place where things are going to change. I think the other places where it's going to change, which isn't really technology, I mentioned that one of the key factors when you're thinking about pre-implantation genetic testing up until very recently is you have to perform embryo biopsy, meaning that somebody, an embryologist, it's a very special biologist that understands how embryos live and breathe. An embryologist can learn how to use a laser to cut a small hole in the protein case that surrounds an embryo called the zona pellucida and carefully remove a few cells for testing, leaving the embryo perfectly intact. And it looks awful when you see it. If you go on YouTube and look up embryo biopsy, it looks amazingly awful, and you can't imagine any embryo could ever survive, but they do. But that takes technology. It takes lasers. It takes special microscopes. It takes a lot of training. And not every territory in the world has hundreds of embryologists or thousands of embryologists like we have here in the States and like you have in Australia. Not everyone has the ability or has that technology at their fingertips. So not unlike non-invasive prenatal testing, NIPT or NIPS, which I'm sure you've covered in your podcast before, it turns out that not only do fetuses spill DNA into the human bloodstream that one can use by taking a non-invasive blood screen to look at the fetus, it turns out that blastocysts, a certain kind of embryo or a certain stage of embryo development, they spill DNA into the culture media. And so the absolute newest thing in the field right now is what we're calling non-invasive prenatal genetic testing. So the ability to sample the culture media that the embryo's been living in that has cell-free DNA in it, and to be able to then assess the embryo for, we think, all the things that I mentioned: structural chromosome rearrangements, single gene defects. Right now, we've tested, certainly there's some papers out there that have tested pre-implantation genetic testing for aneuploidy on spent culture media. There's a couple of clinical trials going on right now. One would imagine, I have a student that's working on this, that ultimately we should be able to look at structural rearrangements and monogenic diseases in the culture media as well, which will do two things. One is it opens the technology into territories that don't have embryologists that can just do this anytime they want. Two, it should reduce cost, because obviously cutting an embryo and that technology costs money. So this should really, the non-invasive should really open this test up to a whole new world of people that don't have access to that test today, and therefore are simply choosing the best embryo to transfer simply by looking down the microscope and saying, I think that embryo looks best, which is not ideal.
Far from it. And technology never ceases to amaze me, Gary. I mean, this is such an amazing development in this part of technology. I can imagine that this will simplify, it'll reduce cost, it'll improve outcomes. And really, I mean, that's the point of this, isn't it? That's the point of companies like yours, of the scientists and the medical technologists working on these kinds of technologies so that we can improve human health. So, and I guess when do you expect these kinds of new technologies to be implemented?
Well, certainly, as I mentioned, there are two groups. One of them is in Australia that's currently offering non-invasive prenatal pre-implantation genetic testing for aneuploidy. They're offering that clinically in their IVF center. They're the only IVF center in the world that I know that's offering this directly. There is a Chinese company that has a service, and they're out in the field selling that service. And then, as I mentioned, one of the big genetics companies has just released a kit that then anybody that owns a next-gen sequencing system and understands pre-implantation genetics, anybody that offers that test today on invasive biopsy can offer that test in a non-invasive way using this new kit that's just been released. It's been released literally a month ago. So
so this is really new.
It's bleeding edge.
Bleeding
I think the miniaturization, from the sequencers, they've been miniaturized. Certainly, again, one of the big sequencing companies has made some smaller sequencers that quite nicely run 8, 10, 12, 20 samples. I think the library prep automation, I think that's a little bit farther behind, but I'd say probably in the next 18 months to 2 years, I think you're going to see things like that really hit the market and work well.
Great. Well, it's been very illuminating, Gary, and excellent for me to hear about, and I'm sure our listeners to hear about, implantation genetic testing and some of the really great new technology coming along. So it just remains for me to thank you very much for spending some time with us, and draw a close to this podcast. Be listening out for other podcasts on this and other topics coming out in the coming months. Thanks very much, and bye for now. Thank you for listening to this edition of It's in Our Blood. Look out for further topics in this series, or go to pathologytechnology.org.au. This podcast is produced by London Agency, Melbourne, Australia.